Hi everyone. Today we're presenting on the effects of alcohol and NyQuil on the dopamine system of Drosophila and corresponding sexual and aggressive behaviors. These experiments were done by Mariano Spanos with help from Jess Bohan and Peter Casey Kaplan. For this lab, we aim to discover the effects of alcohol and NyQuil exposure on the dopaminergic pathway of Drosophila melanogaster in relationship with the courtship and aggressive behaviors. Can you go to the next slide, please? Thank you. First, for health relevance, some background. There has been increased recognition of the abuse of over-the-counter medications in public health circles, such as the common cold and flu medication NyQuil. NyQuil is a drug that contains two very strong compounds, which are acetaminophen and dextromethorphan. When these are taken in large amounts with alcohol, they can have detrimental and sometimes deadly effects. These effects include slowed breathing, impaired motor control, behavioral changes, and memory impairment. Despite the major impact NyQuil abuse has, specifically the simultaneous use of NyQuil and alcohol, little research has been conducted on the effects of this abuse on social behavior. As a result, we aim to analyze the effects of NyQuil and alcohol exposure on the dopaminergic pathway and look at fly social behavior. Next, we want to discuss why we're using flies. So fruit flies were used in this experiment due to their easy accessibility and the conservation of complex circuits and neurons between the fly and human brains. Additionally, the genome of Drosophila melanogaster has been fully sequenced, allowing for easy manipulation of the genome to express desired traits. For our flies, we are using halorhodopsin and channelrhodopsin. So we're looking at the dopaminergic pathway within the flies. This is the pathway of interest since dopamine regulates behavioral activity similar to that in humans, such as arousal, social behavior, courtship, and aggression. This allows us to regulate the pathway while observing the direct impact that alcohol and NyQuil exposure has on these social behaviors. As for why alcohol and NyQuil, can we go to the next slide? Thank you. Why alcohol and NyQuil? So we're using 70 proof ethanol for the alcohol, and we're exposing it to flies with over-the-counter NyQuil. The effects of alcohol can change social behavior and delay reflexes. We expected to see hypersexuality and hyperaggressive behavior due to the alcohol stimulating the dopamine pathway. As for NyQuil, we expected the two major compounds should cause impairment when abused, and the effects of these compounds would include decreased motor control, slowed breathing, issues with behavior, and loss of memory. We expected to see a reduced amount of social behavior, but not complete cessation due to the sedative effects of this combination that would, however, potentially be rescued by the activation of the dopamine pathway with the channelrhodopsin on switch.
Next slide, please. So we will now be talking about the experimental design. Fruit flies were raised in small vials with Drosophila food mix consisting of yeast and were incubated in room temperature with 60% humidity. Flies were separated based off fly lines and eventually were separated based on sex to maintain sexual naivety. 3 fly crosses were made in this experiment. In order to map the dopamine pathway in Drosophila melanogaster, the fly line GAL4-8848, which is the driving line and targets dopaminergic neurons, were crossed with a UAS 8515 enhanced GFP green fluorescent protein fly line in order to produce dopamine neurons tagged with GFP. This cross allowed for the dopamine neurons in the fruit fly to be detected by green fluorescence, resulting in easier mapping and detection. The progeny of this fly cross was dissected and mapped through microscopy. In order to control the dopamine pathway of Drosophila melanogaster, optogenetic techniques had to be implemented through fly crosses. 2 fly crosses were produced expressing optogenic modifications. One expressed channelrhodopsin and the other line expressed halorhodopsin. The GAL4 driving line 8848 was crossed with the UAS 55134 line, producing progeny containing channelrhodopsin tagged dopaminergic neurons. This cross allowed the dopamine pathway to be activated using red LED light. The GAL4 driving line 8848 was also crossed with the UAS 41753 line, resulting in progeny flies containing halorhodopsin tagged dopaminergic neurons, which allowed for the inactivation of the dopamine pathway when exposed to green LED light. To observe the results of the effects of alcohol and NyQuil on the dopamine pathway in social behavior, 6 behavioral groups were formed. The channelrhodopsin tagged flies were exposed to either ethanol, and NyQuil together, remained as the control group, otherwise known as the wild type group. The halorhodopsin tagged flies were also exposed to ethanol, and NyQuil together, remained as the control group. Fighting chambers were used to confine the flies into a small space with competitive source to capture social behavior fly groups. The chamber was made out of a clear petri dish with glass microscope slides as walls. It was important to make sure that the chamber was clear and that the flies were visible in order to record behavior as well as apply optogenic techniques to the flies. The exposed flies were placed into fighting chambers with a fly from the same exposure group and optogenic tag. A red light was placed to activate the dopaminergic pathway in channelrhodopsin tagged flies, and a green light was flashed to inactivate the dopaminergic pathway in flies. The next slide, please. Flies were anesthetized briefly using CO2 gas during experimental trials in order to restrain from impairing social behavior. The behavior of flies was characterized based on courtship and aggression and were scored based on scoring guidelines from Kravitz et al. 2002. Behaviors included orientation, chasing, tapping, tasting or licking, singing, and mounting. The behavior scoring from the exposure groups were compared to the scoring of the wild type groups, who were not exposed to alcohol or NyQuil.
Okay, moving on to data analysis. So in terms of imaging, the dopamine pathway was mapped through the GALP4 UAS system by targeting the dopamine pathway with GFP. So figure A is a GFP image of dopamine, and figure B is a DAPI stain of the dopamine pathway. In terms of behavioral data collection and analysis, videos were recorded of each trial in lab, and subsequently we quantified the occurrence of each behavior. So the red video is a channelrhodopsin cross of non-exposed flies, and this is a mating chamber. And the green video is another mating chamber, but this is the halorhodopsin cross, again, non-exposed. So there is some mating behavior that was observed in the channelrhodopsin. And there also is some courtship behavior that was observed in the halorhodopsin as well, which is the off switch. Overall, no fighting behaviors occurred in any chamber, but there was mating in both groups for the non-exposed flies. There was no significant difference between the frequencies of mating between the halorhodopsin and channelrhodopsin crosses, which is not particularly consistent with our initial hypothesis that the on switch would induce hypersexuality, hyperaggression, and overall increased social behavior due to stimulation of the dopamine pathway. Moving on to the exposure groups. One sec. So these are the ethanol NyQuil exposed flies in their mating chamber. And as you can see, they're fully sedated and sleeping. And in all our other trials with ethanol exposure and alcohol and NyQuil exposure for both the mating and the fighting chambers, there was no social behavior observed or recorded. Just complete sedation in the ethanol NyQuil exposed flies and just disorientation the ethanol exposed flies. Some more of the sleeping here. So our final results and conclusions, the ethanol and NyQuil exposed flies were unresponsive for all behavioral assays and trials. This was inconsistent with what we theorized would happen, which was that the alcohol NyQuil Chandler Dobson crossed flies would certainly have less sexual and aggressive activity than non-exposure flies, and especially the ethanol exposed flies, which were expected to be hypersexual and hyperaggressive. But instead, we saw no response to the on switch and a complete sedation. So it could be inferred that the activation of the dopamine pathway with the on switch was not enough to rescue the flies from the sedative effects of the alcohol NyQuil exposure. However, in other trials, we also did not observe any behavioral response from flies that were purely exposed to ethanol 2, and as such, repetition of our experiments would be required before we can make any concrete conclusion what the effect of ethanol and NyQuil has on fly social behavior. In terms of future studies and research, we could look into whether observed effects are exacerbated in later generations. To do this, that would require us to very closely monitor each generation of flies, which we did not do in this experiment. We used several different generations of flies to record the experiments. And this could have been a confounding variable in our methods. We can also look into effects of alcohol and other over-the-counter medications on the dopamine pathway. That would be interesting to observe, as alcohol is known to interact with several different over-the-counter medications besides NyQuil. And that's all.